Hi, my name is Margaret Frank, and I'm a co-PI in CROPPS and research lead for Exemplar 1, where we're building living reporters for nitrogen sensing. Plants are wired with internal communication systems that help them navigate complex, changing environments. A founding principle in CROPPS is to illuminate these hidden conversations to enhance resource management in agriculture. Here in Exemplar 1, we have developed programmable plants that communicate their nitrogen status. We have embedded these plants into the essential hardware and software context to enable a new living sensor-based technology for nitrogen management. These plants enable an entirely new approach to nitrogen use based on rapid, living nitrogen reporting. This technology solves Mario's problem and the larger issue of resource management for the rapidly growing industry of controlled environmental agriculture. You'll see at the end of this presentation that this technology also links to promising directions in complex outdoor agricultural settings. These next slides will focus on how we built the programmable plant that enables this new approach to resource management. Plants forage for nutrients in heterogeneous environments. For nitrogen foraging, they express short 15 amino acid CEP peptides in areas of low nitrogen. These peptides travel through the xylem into the shoot system, where they are bound by a CEP receptor, which in turn produces another mobile peptide called CEP downstream, or CEP-D for short. CEP-D moves from the shoot back into the root, where it upregulates nitrate uptake transporters and promotes root branching in areas of high nitrogen, while repressing nitrogen foraging in areas of low nitrogen. We have taken the core components of the CEP signaling pathway and identified related homologs in our target crop species. Here I'm showing you just a small snapshot of our foundational discovery data for CEP peptide function in tomato, where we pulled out CEP sequences, analyzed their evolutionary relationships relative to other eudicot crops, compared their conserved amino acid motifs, where you can see that the tomato CEPs have considerable sequence divergence in the amino terminus relative to Arabidopsis, indicating unique evolutionary histories in these two species. While we annotated 21 CEP encoding genes in tomato, we need to use expression data to select for root-expressed CEPs and shoot-expressed CEP-D paralogs, which are most likely to play a role in this root-to-shoot communication pathway. To test whether these CEPs engage in signaling, we performed peptide application assays where we quantified root architecture responses and modeled for receptor-ligand binding interactions using AlphaFold. It's important to point out that the evolutionary history of this pathway is not perfectly straightforward. For example, in the CEP downstream clade, there have been several independent duplication events, which means that the functional role of CEP-D1 and CEP-D2 in Arabidopsis does not necessarily line up with the two CEP-D paralogs, CEP-D alpha and CEP-D beta, that we found in the reference tomato genome. I'm going to show you why this is important in just a minute. In order to visualize when CEP signaling is occurring and therefore project native root sensing of low nitrogen conditions into the plant canopy, we linked the CEP-D promoter to Ruby, a reporter gene that produces a red pigment when it's expressed. We did this for each of the two CEP-D paralogs in tomato. We then performed extensive whole life cycle analysis for multiple independent events for each of the two CEP-D constructs. This is a mugshot showing wild type for context, and this is an event for our CEP-D alpha construct, where you can see that Ruby is highly expressed in all tissues throughout the life cycle. This was consistent across several events, indicating that this is true to the expression of this gene. In contrast, CEP-D beta, another paralog, is localized to vasculature and is very lowly expressed under full nutrient conditions. We carried multiple independent events of CEP-D beta forward to test for the nitrogen responsiveness of this gene. This was one of our first looks at the CEP-D beta Ruby line, reporting under low nitrogen conditions. You can see that the plants are mostly green pre-treatment and progressively turn deeper shades of red under low nitrogen conditions. To fully understand the functional relevance of these plants, we have embedded them into an automated imaging setup called the FarmBot system, where we are growing the reporters under hydroponic conditions that mirror controlled environmental agricultural production settings and collecting image data in real time. Here's a quantitative look at two separate events of CEP-D beta, Ruby, the bottom two plants in these pots, versus wild type on top. The top row of these images is showing digital images, and the bottom row shows green to red channel from our image processing pipeline. You can see the plants responding to low nitrogen treatment within 24 to 48 hours of treatment. This is several days before traditional nitrogen sensing would pick up on the deficiency. And you can also see the plants responding to recovery within 3 days of returning to high nitrogen, which shows that these plants could be used through continuous management cycles that would be typical in a controlled environment setting. Here is a quantitative look at that data, showing our reporter performance in red versus wild-type plants in green, where you can clearly see reporting turning on within 24 hours and increasing over time, and likewise, reporter turns off under nitrogen recovery. To fully embed our programmable plants into a closed-loop management system, we gathered for a team science sprint this past January. Our team brought together trainees from different disciplines and institutes from across CROPPS to coordinate closing the loop for our greenhouse ruby plants, using living reporter plants to signal when nitrogen is low and automatically adjusting the status of nitrogen in the irrigation system based on the plant signal. The feedback from this CROPPS team was super enthusiastic. In a post-sprint survey, almost all attendees stated that participating in the sprint deepened their understanding of the nitrogen reporter plants from an interdisciplinary perspective, and they were able to see the value of their skills being used across disciplines. Next, I will show you a short footage from a demonstration to local stakeholders that we did at the end of the sprint.
We have built an image analysis tool to actually take these images in the back end that Charnouay was talking about to actually run that analysis in an automated way. And then in the back end, there is a program that produces two different images. One is a segmented image, which looks like this. So what is segmentation? Segmentation is supposed to look at which parts in the picture is plant. Is it plant? Those pixels should show as white, and the background should show as black. After we have a segmented image, let's X out of this. Then we look at how red that plant is. So how many red pixels? This is one of our red alert plants. We can see the redness of it highlighted here. And so then there is a decision-making process that's also in the back end that thresholds on how red it is. So the magnitude of red is a lot higher under nitrogen stress.
We have a hydroponic system to feed the plants with the option for two different nitrogen supplies. We have the robot system to image the plants, and we have the automated analysis pipeline. From there, the next step is to take the results of that analysis so we can see, the plants that are alerting are showing up red. And to then close the loop, all we would need to do is take the results of that, say maybe you set a threshold for the number of plants alerting that you want or a certain proportion, and have the software automatically change the nitrogen supply.
In order to communicate these valves with the decision from the image analysis, there's a microcontroller here, an ESP microcontroller, that talks with the output from the backend. But in a red alert event, that's when the image picks up a plant that is nitrogen deficient. Then the backend is going to recognize that plant and that line needs some nitrogen, and is going to activate, let's just do the C+ line. And so that activates, and we have this red alert.
Awesome. So as you saw from the demonstration, we have built a dashboard that we can use to remotely view the experiment and allows the experimenter, farmer, or grower to adjust the nitrogen fertigation based on Ruby, or to set the adjustment in fertigation to be automatic based on the plant sensors. This is an experiment that's happening now in the Cornell greenhouses, and I could adjust fertigation based on my plant's red values remotely from this dashboard. Now, Professor Hakim Weatherspoon is going to take you behind the scenes of how we have closed the loop for these reporters.
Hi, my name is Hakim Weatherspoon. I'm a professor at Cornell University in computer science with expertise in computer systems and networking. I'm excited to take a look under the hood and see how we have closed the loop and advanced the state-of-the-art not just in biology and agriculture, but in computing and artificial intelligence-enabled decision support. Behind the dashboard is a complete closed-loop pipeline. First, a robot captures images of the plants. Those images are stored in a database, then passed into our analytics pipeline, where we segment the plant images, extract signal values, and determine whether the plants indicate nitrogen deficiency or sufficiency. Those analytics are trained and calibrated so the system can move from raw images to actionable decisions. If intervention is needed, the system can trigger the appropriate responses, including adjusting water and nitrogen delivery. The system can operate automatically based on plant signals and decision rules that we define, or the grower can remain in the loop, setting weekly and monthly schedules. Lastly, there is a live feed where robot and plants can be observed in real time. What is important here is imaging, analysis, visualization, and irrigation are no longer separate pieces. They work together as one integrated system. This is what it means to close the loop. Plants report their condition, computation interprets that signal, and the system responds in a timely manner. We also support flexible operation. Parts of the pipeline can run locally, close to the greenhouse, and in the cloud. The platform is also remotely accessible through a web interface. That means a researcher, grower, or operator can observe the experiment, review the analytics, and change the system behavior from anywhere. What excites us most is that this design generalizes. We can deploy the same approach in a greenhouse today and extend it to field settings and multi-location agricultural systems tomorrow. That is why we describe this as moving toward a software-defined farm. Agricultural infrastructure that can be monitored, updated, coordinated, and controlled through integrated computation and networking. Just as importantly, we are building this with real-world constraints in mind, including intermittent infrastructure and grower data privacy. Moreover, our work has produced a strong paper trail with publications in premier computer science journals spanning computer systems, networking, and human-centered computing designs. So the contribution here is broader than just one dashboard or one experiment. We are establishing the computing foundation for scalable, privacy-aware, closed-loop programmable plant systems.
From this first generation of Ruby reporters, we have started to learn from these plants and found routes to design second-generation reporters with tighter dynamics. This is showing SEPD expression on the left, where gene expression goes up as nitrogen deprivation progresses and drops with nitrogen recovery. And on the right is a related gene family member in the GRXS family that has contrasting expression, where it is on under normal nitrogen and turned off under low nitrogen. Notably, there are several family members that follow this alternative GRXS expression pattern. We're using these contrasting GRXS gene family members to fine-tune the nitrogen reporter dial using dual fluorescent outputs. We are also building on SEPD reporting as a platform for reporting on flexible inputs. Here we have built out prototypes in Arabidopsis with drought-activated root-to-shoot signaling that turn on SEPD reporters and grafted scions. We are now extending this technology into rewired tomato signaling systems. A key question that repeatedly comes up from our stakeholders is when will this be available in maize, where the economic and ecological significance of nitrogen management is tremendous. We have established the groundwork to assess feasibility of SEPD-based nitrogen management in maize. This is a gene tree for the SEPD clade in Ku dicot and monocot crops. Functionally characterized Arabidopsis SEPDs are located in the B gamma clade, this teal clade here. Maize SEPDs are lacking from this clade and are instead found in the sister clade B beta, indicating that similar to our work in tomato, foundational data is going to be essential for engineering a functional route for SEP-based nitrogen monitoring in maize. We have started this analysis by investigating nitrogen responsiveness in SEPD-related maize paralogs, and here you can see some promising leads that set the basis for our future work on engineering nitrogen sensors in maize. I'll now hand you over to Mike Gore, who has led the evaluation of our nitrogen reporter plants in the field with amazing results.
Hi, I'm Mike Gore, Associate Director of Research Partnerships. At CROPPS, we are developing tools to help farmers better manage key inputs. CROPPS has already successfully tested and characterized its root-to-shoot nitrogen reporters in the field, enabling row crop farmers to close the loop and achieve right place, right time management of nitrogen and other key inputs. Established in 1876, the Mora plots at the University of Illinois, Urbana-Champaign, are North America's oldest agricultural experiment field. And nearly 150 years later, the same soil probe is still used. The world is long overdue for a field-based nitrogen sensor, and nitrogen reporter plants are finally making it possible. So how can nitrogen reporter plants improve crop management? Kelly Robbins and his lab are exploring this by simulating nitrogen field maps from real observations. The far left heat map panel shows the simulated nitrogen pattern. The center shows readings from sparsely and randomly planted reporters. And the far right shows the interpolated full map. Covering just 10 to 15% of a field with sentinel reporter plants was enough to reconstruct a full nitrogen map. This work provides a blueprint for a living nitrogen sensor network to guide in-season nutrient management across the field. I began working on programmable plants 25 years ago at Roman Haas Company, when many ideas were beyond the reach of the science. Today, CROPPS technologies have made them possible, and in just 5 years, the project produced a field-ready nitrogen sensor tomato. Our multi-institution team conducted a field trial at the University of Arizona Research Farm in Maricopa, Arizona, demonstrating a functional prototype. Using a vegetation index derived from aerial imagery, purple, red, septi, ruby reporter tomato plants were clearly distinguished from green wild-type tomato plants, confirming qualitative detection. The next step is to test whether the tomato reporter can differentiate nitrogen treatments in the field. Building on the Arizona trial, we tested nitrogen reporter and wild-type tomato plants under standard and low nitrogen at Cornell's Musgrave Research Farm in Aurora, New York. Using the same vegetation index developed for aerial imagery as in Arizona, we provide quantitative evidence that septi ruby reporter tomato plants exhibited significantly greater redness under low nitrogen, reflecting nitrogen stress, whereas wild tomato controls showed no change. For reference, in the image on the left, the corn border rows surrounding the tomato plants, included to satisfy APHIS requirements, also displayed yellowing under low nitrogen conditions. These first-year results from Arizona and New York tomato fields show a key step towards a living nitrogen sensor network, with these sentinel plants dynamically reporting soil nitrogen to guide management, reduce waste, and boost crop performance and sustainability. In July 2025, CROPPS showcased its nitrogen reporter plant research at Cornell's Aurora Farm Field Day. This field day was attended by 200 farmers, educators, researchers, and students. CROPPS trainees presented on first-generation reporter prototypes tested under varying nitrogen conditions, allowing the team to demonstrate the system in real field settings and gather feedback. Stakeholder questions focused on using the reporter in corn and other crops, the benefits of an early warning nitrogen system, cost and accessibility of the sentinel plants, and drone monitoring frequency. The event demonstrated strong interest and provided valuable guidance for developing and deploying nitrogen reporter plants. In summary, we have closed the loop on a programmable plant system for nitrogen management. Septi ruby reporter plants have shown proven responses to nitrogen depletion and recovery, and an integrated decision support system for fertigation adjustments has been demonstrated in the greenhouse. We are on track to achieve full field implementation in 2026-27. This example uses conventional and robotic actuation of fertigation, but next, Tony and Arjun will present a case of programmable plant-based actuation. Thank you.
Hello, my name is Tony Studer. I'm an associate professor in the Department of Crop Sciences at the University of Illinois, and today I'm a single person representing the many scientists working on Exemplar 2, programming plant architecture. As you heard from Abe earlier, programming plants has the potential to greatly impact agricultural production and sustainability. The figure in the top left here from Bayer crop scientists and with feedback from stakeholders, I've really been driven to think about the crops of the future and what we could do with programmable plants. Plant architecture changes played a significant role in the Green Revolution and continue to increase yields even through decades of breeding. This is especially true for leaf angle, which has been a major trait for optimizing maize for higher planting densities. I'm going to tell you a story about what we've accomplished towards programming plant architecture in maize, specifically with leaf area, and our plans for future innovations in this field. But before we get into the specifics, it's important to realize that our vision of programmable plant architecture requires three main components, namely an understanding of the genes that control our traits of interest, a control system by which we can dynamically tune expression of those genes, and models to help guide how we alter gene expression to reach our ultimate outcome. As such, this requires bringing together expertise from different disciplines, such as genetics, synbio, plant developmental biology, crop modeling, agronomy, and others. And this is only possible thanks to the NSF STC CROPPS. Now to that first component, genetic control. The gene that we've been studying in maize is called reduced leaf area, or RDLA. As you can see in the picture on the left, wild-type plants are much leafier than mutant plants on the right that have the same genetic background. We've identified the gene controlling this trait and characterized it across 27 different elite genetic backgrounds. There's a lot of power in this type of genetic analysis, and it enables us to dissect mechanism, conduct in-field evaluations, and generate commercially relevant hybrids for yield testing. I told you that modeling was another requirement for programming plants. Our phenotypic measurements in these different backgrounds have been used to make some incredible progress on tuning 3D canopy architecture models that can serve as digital twins. Megan Lang can now grow in silico maize plants of different genetic backgrounds with and without the RDLA mutation. Her in silico maize plots can be seen in the photo at the bottom. She has also used the model to measure light interception throughout the canopy, which is shown in the movie and in the still graphic as light interception across the different genetic backgrounds with and without RDLA are shown. What's really exciting for me is her model predictions have started to mirror what we're seeing in the field. Such a digital twin is very powerful when thinking about designing and programming plant architectures. Now let's talk about the real field, where we look at how changes in leaf area can be tuned for optimum productivity. For the past 2 years, we've grown wild-type and RDLA mutant hybrids in large multi-location yield trials that look at genetic modifiers of the RDLA phenotype and its interaction with planting density. This picture at the top left shows our 2025 field and the scale of the research. This trial has 125,000 maize plants in it. In the zoomed-in boxes, you can clearly see the difference between the wild-type and the RDLA mutant hybrids at different planting densities. To study the impact of these leaf area changes on canopy closure and yield, weekly drone flights captured multispectral images of the trial, which are currently being analyzed by the Robbins Lab. So what did the yield data tell us? Well, the biggest surprise to me was that the data revealed that maize hybrids are too leafy, and a reduction in leaf area does not incur a corresponding reduction in yield. We also see different responses to leaf area reduction across different hybrids. That's what's shown in the figure on the left, where we're plotting percent change in leaf area by percent change in dry matter grain weight, or yield. Some hybrids can tolerate a significant reduction in leaf area without a large reduction in grain yield. However, it's important to note that this is not a uniform response across all backgrounds. As you can see in this figure, where we're again plotting percent changes in leaf area reduction, you have different genetic backgrounds responding differently at various spots in the canopy. Although we modulate percent leaf area reductions in these different genetic backgrounds, it's important to note that we can only make modest changes to canopy shape, which is plotted over here. In this final graph, I'm showing absolute leaf area, and you can see that the widest leaves are always in the middle position. This is not only something that we've observed. In a paper recently published by scientists at Bayer, they looked at hybrids across a 30-year time frame and found that, quote, on a per plant basis, breeding did not affect plant leaf area or its distribution. These are interesting observations and suggest that there is a need to program architecture to break natural constraints surrounding this trait.
Hi, my name's Arjun, and I'm going to build on Tony's description of our progress in maize by giving you a summary of work we've been carrying out in tandem to create programmable architectures in tomato. In contrast to the top-down breeding-based approach to engineer architecture that Tony told you about in maize, our approach in tomato is based on bottom-up design that uses mathematical models to guide the deployment of synthetic transcription factors that modulate developmental pathways. Specifically, we're modulating gibberellin signaling to restrict the growth of the shoot, to enable more efficient carbon partitioning, and to expand the root network to increase nitrogen and water use efficiency. This kind of modulation has historically resulted in dramatic yield increases, as evidenced by the Green Revolution varieties of cereals, and our approach to implementing it has the potential to achieve similar gains in tomatoes while minimizing the trade-offs those varieties had in terms of high input demands. This effort builds on decades of work that have been done to understand the role of gibberellin signaling on the regulation of organ size in plants. This pathway has multiple layers of feedback, as can be seen from the complicated diagram on the left, which makes it challenging to intuitively predict how changes to the expression of genes in this network would impact plant growth. To overcome this challenge, we built a modified version of previously developed ordinary differential equation-based models of this pathway, and this model predicted that modulating expression of the gibberellin receptor GID1 would enable efficient and dose-dependent control of organ size. To test this prediction, we built stable lines of the model plant Arabidopsis thaliana that expressed repressors targeted to the promoter of each GID1 gene. We then phenotyped these plants, and we saw significant dwarfing in the roots as predicted by the model, and that the strength of regulation controlled the magnitude of organ size change. Excitingly, this model not only enabled the comparison of different regulatory architectures to identify optimal control points in this pathway, it was also able to accurately predict the organ size change generated by these perturbations. Not only is this the first demonstration of quantitatively predictive models relating molecular signaling to whole organism development to our knowledge, it demonstrates a framework for the bottom-up design of plant morphology. Because this model was built using data from roots of Arabidopsis, we were interested in understanding if its predictive power would hold across other tissues in the plant. So we phenotyped various shoot tissues from these plants, starting with hypocotyl length, then rosette area, and then leaf area. And as you can see, we observed consistent dwarfing effects across various tissues and developmental time points. All the work that I've told you about so far was done in Arabidopsis, but our goal is to re-engineer morphology in tomatoes. So we next tested if these models' insights could be generalized to tomato. Phenotyping of multiple independent transgenics that had synthetic repressors targeted to the GID1 genes in tomato demonstrated a consistent dwarfing phenotype across both the roots and hypocotyls. So while it's exciting that GID1 seems to be a generalizable control node for organ size, all the work that I told you about so far has been in pretty controlled lab settings. Thus, it was important to test if these phenotypes stayed consistent in agriculturally relevant environments. So we used our line with the strongest phenotype and grew it in a greenhouse trial lacking both light and temperature control to test if this was the case. Excitingly, when we measured hypocotyl and epicotyl length, we see a consistent dwarf phenotype in fluctuating environments. This demonstrates that in modulating GID1 expression, we have a reliable way to engineer organ size in tomato. This bottom-up design of crops using first principles is a radical departure from the traditional top-down selection-based approaches and could dramatically accelerate crop improvement. This work was recently published in PNAS if you'd like to learn more. We are now working to generate tomato lines that have differential regulation of GID1 in shoots and roots to enable shoot dwarfing and root expansion, and to build quantitatively predictive models for each of these tissues. These lines will be crossed against a pan-genome population and tested in the field by Kelly Robbins' group in the center to collect data for field-scale yield models that incorporate information about genetic background, environment, and management. Our ultimate goal is to connect our mechanistic models of phenotype with correlative yield models to create a new class of predictive hybrid model to help guide how to best reprogram morphology to enhance yields in the field. Kelly's group has developed computational pipelines using drone-based phenotyping data from tomato trials of diversity panels to enable this work in the coming year. In addition to knowledge of the genetics underlying architecture and the predictive models that can guide its tuning, we need tools to enable closed-loop control that tunes the strength of phenotype to maximize yields for a given environment and management. Our ultimate goal is to connect our mechanistic models of phenotype with correlative yield models to help guide how to best reprogram morphology to enhance yields in the field. Kelly's group has developed computational pipelines using drone-based phenotyping data from tomato trials of diversity panels to enable this work in the coming year. In addition to knowledge of the genetics underlying architecture and the predictive models that can guide its tuning, we need tools to enable closed-loop control that maximize yields for a given environment and management. To enable this, we have created a novel Cas9-based synthetic transcription factor system whose activity can be induced using commercially used insecticides called methoxyfenazide, which is sold by Corteva as Intrepid. This chemical is rated safe for use in field settings and is formulated specifically for systemic uptake in plants and stability in the field, making it an ideal inducer for crop trait engineering. We've characterized the system by transient expression, and by comparing the middle two box plots on this slide, you can see that in the presence of inducer, we see significant activation of gene expression, demonstrating that the system was functional. We've also demonstrated that the system can be coupled to GID1 repression in Arabidopsis to enable inducible dwarfing of plants, as can be seen from the data on the right. We're now working with Mark Palstein's group in the center to integrate these lines with their nitrogen reporter lines to enable a demonstration of closed-loop control of architecture in Arabidopsis in the coming months. In phase 2, we will be able to quickly bring together the plant as a reporter capabilities that Margaret showed you with programmable changes in architecture and tomato to create a CROPPS-enabled breakthrough in plant-informed modulation of a critical complex trait. So to quickly summarize the progress that Tony and I just covered, we have identified key control genes for architectural improvements in tomato and maize. We've developed models to connect molecular signaling to physiology and physiology to yield, and we have developed agrochemical inducible control systems. We're now working to integrate these models across scales, to couple the inducible control systems with architectural control genes in tomato and in maize, and to perform proof of concept closed-loop control of architecture in Arabidopsis. Thus, our work across maize and tomato has established the tools and insights needed to realize this vision of closed-loop control of architecture in the coming years. Our work was driven by the social scientists in the center who engaged with growers and demonstrated that what they were most excited about were these kinds of architectural control genes. And it has significant potential for broader impact via its capacity to be translated by companies into products, as evidenced by the in-kind support that Bayer has provided to facilitate this work.
Hello, I am Jesse Woodson, Associate Professor at the School of Plant Sciences at the University of Arizona, and I'm going to be telling you about Exemplar 3, which builds on Exemplars 1 and 2 by combining plants as reporters and as responders using systemic acquired acclimation, or SAA signals. An original vision within CROPPS was to open a dialogue with plants, and to us that means having a two-way communication, that is, the plant speaking to us and us being able to speak to the plant. And we want to achieve such bidirectional communication, taking advantage of the electrochemical and hydraulic waves plants use to naturally communicate messages about its environment. These are called systemic acquired acclimation, SAA signals, and involve small molecules like reactive oxygen species and calcium ions, which locally accumulate at stress tissues and then activate long-distance waves to distal tissue to potentiate changes in stress physiology. And this can allow for a root to shoot and leaf-to-leaf communication. So to build out on Exemplars 1 and 2, CROPPS wants to harness these signals to open bidirectional communication. And that means plant-to-person or reporter plants, where we can visualize these waves in real time to understand how plants are responding to their environment, and person-to-plant or responder plants, where we can potentiate the initiation of these signals to drive physiological changes from a distance. Now, in general, responses to the abiotic environment are going to be very rapid, within minutes, because these are going to be changing very quickly. And harnessing wave signals will allow us to open two-way communication about the daily stresses plants encounter. And this is important whether you're a corn maize plant growing in Illinois or in Maricopa County outside of Phoenix. These are two completely different environments, but one thing they have in common is that each day is a little bit different. Different stresses the plants are going to encounter in terms of water availability, humidity, and temperature. And so by targeting this different category of plant responses, that is water stress, heat, and wounding, we really differentiate ourselves from Exemplars 1 and 2 and open up the ability to create programmable plant systems responding to a different class of stresses and responses. Now, because of the complexity of long-distance signaling and the genetic tools and synthetic biology required to achieve this. We are first working in model organisms like Arabidopsis and Nicotiana benthamiana to be able to establish the technology and provide a proof of concept. At the same time, we are moving towards tomato and envision tomato as the crop plant of which Exemplar 3 will be used in practice. Now, to program two-way communication with plants, we need to do several things. We need a deep understanding of the long-distance signaling mechanisms plants use to coordinate whole-body physiology with their environment. We need the ability to monitor and visualize these signals in real time, the ability to control and potentiate these signals at will, and to close the loop with an automated system that responds to plant signals. Now, like Exemplars 1 and 2, I'm showing you the reporting, modeling, responding wheel as we close the loop in Exemplar 3. In terms of reporting, we are using detection of waves with calcium reporters like our gecko, which responds to different types of physiological stresses. I'm showing you Arabidopsis, our gecko plant, responding to a wound stress. You're going to be seeing this a lot. We also have this in tomato that will allow us to monitor these waves in that crop. As we monitor these waves, we'll be able to model how their waveform correlates to different types of physiology and tell us the biological meaning behind those waves. We're also incorporating this in demonstration space called CROPPS in a Box, which I'll be telling you about later. With responding, we are incorporating synthetic biology tools in the form of optogenetic switches that we can use to non-invasively induce these long-distance signals to potentiate changes in stress physiology like stomatal aperture. And we're incorporating automated detection and automated actuation to close the loop in this system. And so I hope you can understand how we see this as a very transdisciplinary exemplar, just like Exemplars 1 and 2, and how such technology could only be achieved with something like an STC. Now, I want to switch how we talk about this a little bit moving forward with an engineer's view of a programmable plant system. So I think thinking about Exemplar 3 in this way actually makes more sense because we're talking about a very complex system that has both reporter plants and responder plants. So you need to think about how the system in a circuit is going to be affected by inputs, disturbances, and actuation, how those lead to different responses, and how feedback can be monitored and fine-tuned. And this is actually how we talk about Exemplar 3 in our working groups. And so I think this is a valuable way to discuss it. So in this case, the system is the plant. The responding is going to be the agronomic traits and physiology that changes in response to the signals that go through the plant. Those signals are going to be controlled by disturbances and inputs into the system, and those disturbances can be caused by environmental stresses like drought and increasing temperature, which are, of course, related. And the response to those stresses in the form of stress responses like changes in gas exchange, water use efficiency, and drought tolerance. So we've been spending a lot of time in phase 1 understanding how such disturbances affect the system and affect those signals that move through the plant to potentiate these changes in physiology. Last year, for example, we've published an exciting paper in PNAS where we investigated how such disturbances lead to hydraulic waves that rapidly move through plants to potentiate changes in stress physiology. In a paper that is being prepared for submission, we also have looked at how different temperatures can lead to different types of waveforms that travel through the plant, and that plants have quantitatively and qualitatively distinct waveforms in response to different temperatures, not just cold versus heat, but different levels of heat lead to distinct waveforms that we think can code valuable information for the plant to allow it to make informed decisions about its stress physiology. For feedback, we're monitoring the visualizable reporters like our gecko, which fluoresces in the presence of calcium. But at the same time, we are using gene expression as a way to monitor these long-distance waves because they also can control gene expression both locally and distally. For instance, STAT10 is a stress-induced gene that's induced by heat and locally treated tissue over time, but it's also induced long distance by the signals generated by heat. And so this is gene expression controlled by the SAA waves. And this is not a generic wave that's just induced by all different types of stresses leading to the same responses. That does not seem to be the case. Instead, there's specific information leading to the induction of specific gene patterns. For instance, ZAT12 I showed you is induced locally and distally by heat, but it's not induced by cold. Instead, cold induces ELIP2, both locally and distally, but ELIP2 is not so much controlled by heat. So this is meaningful in two ways. In one, it shows that there's distinct information encoded in these ways, leading to different changes in gene expression distally. At the same time, these could be valuable tools to create reporter plants where we can visualize how plants respond to heat and cold, perhaps simultaneously. And so we're using the promoters of these genes to drive the expression of visualizable reporters in phase 2. In addition to calcium, reactive oxygen species has been shown to be a very important part of long-distance signaling in plants, and we have a genetically encoded hydrogen peroxide reporter called HYPER7. It's been shown by other groups that the level of hydrogen peroxide needed to induce these long-distance signaling is not cytotoxic. It's a physiological level that has been shown to play a role in signaling in cellular systems. So this led us to hypothesize that perhaps we can control some of these long-distance changes with ROS, like hydrogen peroxide. So simply just adding hydrogen peroxide to an Arabidopsis root does in fact lead to a rapid long-distance signal up through the root into the shoot, and we can visualize that in real time within minutes with our gecko. And this is significant for us because we have modeled in a paper that's in revision right now that the accumulation of reactive oxygen species like hydrogen peroxide in stomate cells is a potential control point for turgor pressure and the opening and closing of stomates. So we want to be able to connect such ROS being made in the root to a long-distance signal that could potentially affect stomatal regulation and water use efficiency. Of course, adding hydrogen peroxide to roots is not necessarily feasible, particularly in an agronomic setting. So we have also generated optogenetic photosensitizer proteins that can be activated by monochromatic light to produce reactive oxygen species in a non-invasive way. So this is one type of optogenetic switch, if you will. To show this works in vivo in plants, where we can transiently express these ROS tags in Decocciana benthamiana and show a light-dependent induction of reactive oxygen species in a short amount of time. And so we are now testing the ability of that to induce a long-distance signal that could affect stress resilience either through stomatal regulation or gene expression. And so we put these ROS tags in Arabidopsis under the control of a root-specific promoter. So these plants make the ROS tags in the root. They're fluorescent, so you can see that here in yellow at the bottom here. And at the same time, we've driven the expression of these ROS tags under a guard cell-specific promoter, SLAC1, so we can just make ROS specifically in guard cells in a light-dependent manner. We can test our model on stomatal regulation that way as well. We have made other kinds of optogenetic switches in plants. Christian in my group has also created a phytochrome-based optogenetic switch that doesn't make ROS, but just directly regulates gene expression in response to red light. It can also be turned off by far red light. So this works in vivo. Again, in Decocciana benthamiana, he has shown that you can turn the switch on and off when driving a fluorescent protein like YFP. So under far red light, it's off. You don't see any fluorescence. Under red light, it turns on, and now you can see that YFP fluorescence. As a control over here, we have constitutive expression of YFP. So it's not light dependent at all. So that shows that on the bottom left here, that expression of YFP is indeed due to the red light activated switch. And to close the loop, we are also adding automated interactions between plants and robots, both for the detection of the feedback in that loop and also to potentiate the long-distance signals through the activation of these optogenetic switches. So that's what I want to talk about next. It's a closing the loop, and we're doing this in two ways in Exemplar 3. First with CROPPS in the box, and second with FarmBot and TubeBot in the greenhouse in the agronomic setting. So with CROPPS in the box, this is this fantastic intermediate step between the laboratory and the greenhouse where we're able to have a demonstration space of a plant-robot interaction and watch how plants respond to their environment in real time and to be able to interact with that plant. So bidirectional communication. So the way this works is that the robot interacts with the plant to cause a stress like a wound. It has the RGECO reporter in it, so it produces a signal that can be detected by an RGB camera that is then decoded, and then information is sent back to the user about what is happening. So this is how it works when you think about it in terms of the circuit. The user sends a text to the plant to wound it. The plant's wounded, it makes an RGECO signal, which is then decoded by the camera, and an output is sent back to the user in the form of a text message that says, "I'm hurt." And here it is in actual practice with Israel CROPPS postdoc, who has sent a text to the box to tell it to tickle the plant. So a low-grade mechanical stress. The plant responds immediately within seconds by inducing a wave that can be visualized in a couple leaves at a particular intensity. And then that is decoded, and that information is sent back to Israel, and the plant says, "Yes, I was tickled. I felt that." Now, Israel's going to send a second text to the box for a much more stressful wound, which then leads to a quantitatively distinct signal throughout the entire rosette at a very high intensity. That is then caught on the camera and decoded as a wound, and that information sent back to Israel. So this has been a collaboration with IBM, and it's been a fantastic transdisciplinary training experience for our CROPPS trainees, like postdoc Nathaniel, who is a postdoc at University of Arizona and a biologist, but he visited Cornell during a sprint to be an engineer and learn and work with the engineers at CROPPS at Cornell. This has also been a demonstrations tool that we've brought to outreach activities to demonstrate how we can establish bidirectional communication with plants. We brought this to high schools and to university undergraduate courses. And we are working with our computer scientists to find the best ways to collect information, all the rich information we're getting from these crops in the box, and ways to store it for modeling activities for now and in the future. But the vision of crops in the box isn't just to wound a plant and see that response. It's to incorporate different kinds of stresses that will be perceived in different ways by the plant, leading to distinct waveforms, which then can be monitored and decoded. And then that information can be sent back to the user for information about what that plant is experiencing. For instance, we can send a text to the plant for a heat stress that then we know leads to a distinct waveform that can then be visualized and decoded, and then that information can be sent back to the user, and the plant can say, "I'm hot." With that information, the user could then manually dial down the temperature around the plant. Calcium waves would settle down to baseline, and then the plant would send a second text, "Okay, I'm okay now." We're looking into the future. We can do this a different way. We can also have the crops in the box shine light onto the root systems that harbor these optogenetic switches that can induce long-distance signals through the plant to open a stomata, regulate stomates to regulate transpiration, which can cool the plant that way. So we see this as a complete example of closing the loop with Exemplar 3, and something we are moving towards in phase 2. At the same time, we want to be able to take Exemplar 3 to an agronomic setting, to a greenhouse. So this is where we're using FarmBot, which is an automated above-ground canopy imaging system, and TubeBot, a below-ground root architecture imaging system. And they're integrated into a framework allowing for comprehensive automated phenotyping capabilities. This is a collaboration between Cornell University and Tuskegee University, and a good example of technology being developed at one institute and being transferred to another for optimization and use in the field. So here we have the Tuskegee University students were invited to Ithaca to meet with Cornell faculty, where they were learning about the automated plant-robot interactions, which they're going to take back to their campus and use in their greenhouses. So here is that FarmBot and TubeBot in action in a Tuskegee University greenhouse led by Greg Bernard. And here we have the FarmBot able to monitor the tissue in the canopy, and then the TubeBot is literally the bot in the tube moving underground, under the soil, interacting with root systems. So the concept here is very similar to crops in the box, except in this agronomic setting, much more complicated too, of course. But as we envision it, is that the FarmBot is going to be able to collect information about the physiology of the plants and the waveforms moving through them, and the TubeBot is going to be interacting with the roots harboring optogenetic switches, where those can be turned on actuated to induce long-distance signals to potentiate changes in stress physiology in the canopy, which will be monitored by the farm bot. And I hope you agree that, or you can at least appreciate that with this farm bot 2-bot example and the crops in the box example, this is transdisciplinary projects that would never be possible outside of something like an STC that allows scientists of all different types, biologists, engineers, computer scientists, agronomists, to get together and develop these very complex and exciting technologies. Okay, to summarize what I'm telling you about with Exemplar 3 with respect to closing the loop and reporting, modeling, and actuating. With reporting, we've developed a deep mechanistic understanding of long-distance SAA waves and adopted reporters to monitor waves in real time, identified stress-specific wave patterns and gene targets, automated detection in crops in a box. We look forward to automated tracking of water deficit with FarmBot and 2Bot and develop stress-specific visual markers. So the orange indicate what we'll be doing in phase 2. With respect to modeling, we've models to model control and water use efficiency and models to decode waveform patterns. In phase 2, we look forward to modeling to connect waveforms to plant physiology and models to connect above and below ground imaging to actuation of responses. With respect to actuating, we have 2 different classes of optogenic switches that we've developed to drive gene expression and wave initiation, 2 automated systems that will allow for automated induction of waves. And in phase 2, we're most excited for the automated control of long distance physiology with robot and plant interactions. So to conclude and to wrap up, I want to celebrate CROPPS' phase 1 achievement of realizing our bold founding vision for programmable plant systems. Working in tomato and maize, we have pioneered new programmable functionalities across reporting and responding and demonstrated their integration with analysis and automation in agronomically relevant contexts for both controlled environment and field-based production. We have further built an exceptional team and a rich pipeline for discovery and translation into phase 2 and beyond. Thank you for your time and your attention, and we look forward to hosting you virtually at our upcoming site visit.